Yeah, so a long time ago, I was just a scientist rolling the dice on 4 years of my life, hoping the hypothesis I'd chosen would give me something useful at the end of it. I knew what the odds were; they weren't great, but you want to give it a shot. Fast forward to today, you have this amazing pattern recognition, which is machine learning, that can look at things that you wouldn't be able to pick the patterns out yourself. So I just want to describe a success story, and that's going to frame the question for the workshop, which is sort of taking where we are and taking it all the way to the end mission. So just briefly talk about Yamanaka factors. So Shinya Yamanaka was on the search for proteins that could basically turn adult cells into stem cells. He was successful. It's a really great story if you manage to find the chance to read the book. But just describe what's going on. So on my right-hand side, the blue line is epigenetic age, which is quite a robust biomarker of aging, and you can see it going down. And at the time, this was like a massive breakthrough that you could reverse age all the way down to zero. But Shinya Yamanaka was never searching for rejuvenation. He was searching for pluripotency. And one of the qualifying assays of pluripotency is that pluripotent factors induce tumors in animals. So clearly, there's something really exciting, which is the rejuvenation, but a big problem if we just want the rejuvenation, which is these tumors. So maybe we should look a little bit deeper and find out, is rejuvenation a different pathway from pluripotency? Because if it is, we could have more of that and less of the thing we don't want. So given there's 4 transcription factors, which are our current definition of rejuvenation, if we want to explore all other combinations of transcription factors, it's basically impossible in the real world. It would just take centuries, if not millennium, to do the combinatorial space of experiments. But as of today, we do have virtual cells. So they're very narrow virtual cells. But if we just take gene expression, we can take a machine learning model, train it on single cell gene expression data, and it can learn the relationships between the genes to the extent you can sort of abstract a graph of genes and run experiments on that graph. So we use virtual cells, we use aging clocks, which is another product of machine learning, and we run these big campaigns of virtual experiments. So in this case, it's about 5 years of real-world experiments compressed into 1 year. So it's about 350,000 experiments. And we've got the change in the age of the simulation on the bottom, and then we've got the number of experiments that induce that change. So in the green box, rejuvenating gene combinations, and in the red box, pro-aging interventions, which could also be a target for drugs. And the first thing the simulations told us was there might be single genes that are sufficient to do this. So we tested this single gene speculatively. We didn't believe it'd be that easy. And this single gene reversed epigenetic age in real aged fibroblasts in the dish, and it didn't induce pluripotent colonies. So the white dotted line in this diagram is a colony that we pick out. So it sort of blew our mind that just by asking the question, could we rejuvenate, just purely asking that question, we find single genes, or at least machine learning tells us that. Very recently, we also showed this gene can rejuvenate other cell types. So it's not just fibroblasts, not just the system we optimize for. So again, it's this sort of gift of machine learning. It just saw this gene, told us we should do something. We try it, and it's working now in multiple cell types. And just briefly, how do you create a cell aging clock, which was one of the 2 things that we used? A huge data set of single cell transcriptomes, but each with an age label. So you now train machine learning to predict the age based on the gene expression. And then you get a single cell clock. These virtual cells, quite sort of nice graphics, but 60 million single cell transcriptomes. That's what it looks like if you do a UMAP plot. And you apply neural nets to capture the relationships between the genes. And then you put them together. They're both black boxes, so we call it dance of the black boxes. And it delivers the goods. Because the machine learning told us there were these single genes, we then asked the question, maybe there's other single genes, and we did a systematic screen of single genes, and we find lots of single gene targets capable of rejuvenation, at least with our clock, but also aging the system. And you can see where our first gene sits, which is not one of the higher ranking genes. It's actually 46th out of all of them. This is the raw data. So basically, if the cells are getting younger, it's darker. So you see sort of a line around this gene called SP000. If it's yellow, it's getting older. And some of these genes intersect with known lifespan extending pathways to just show you the quality of the gene predictions these sort of machine learning models are creating. And this is just a big thank you to the team, but I just want to frame the problem for the workshop, which is once you find a really great gene, once your wishes are fulfilled by the machine learning genie, how do you turn that into affecting that gene across a whole human body? So what machine learning tools can take us from where we are, which is in the dish, overexpressing a gene, making that younger, to doing that across the whole human body. And I suspect there's lots of different types of tools that come together for that. But I'd be very interested if you've got any ideas. We hive mind it in the workshop. Thank you very much.
All right, I think we may have time for one question. Do we have a question from the audience? Any questions? Where is it?
So when you look at transcriptomics, there's like 20,000 genes, and all of those have some reads. So lots of genes are active all the time. And you do the clocks, you do the epigenetic clocks, you do all kinds of analysis, and always there's a small, very small number of genes that matter. Do you think that's an artifact?
Yes.
Why are we always finding so few genes?
Yeah, so we're not going to second guess it. We're going to keep pushing this stuff into like an organism. Does it work? Does it work functionally? But there are sort of genes that may be used very briefly during your lifetime, and then they're not used anymore, like especially in development. So just something interesting about this single gene we found, specifically not expressed pretty much anywhere in the body apart from the germline, which is very interesting, right? Because the germline's part of the body which has sort of greater protections than the rest, the rest of the body. So I'm trying not to second guess why. I'm letting the machine learning deliver the amazing alien aircraft sort of into the hangar, and then we try and reverse engineer it. Yeah, it's amazing. You can just do a PhD now, and this stuff is like in your toolkit.